Welcome to our first Forefront webinar delivered by dementia researcher in association with the Alzheimer's Society. I'm Adam Smith. I'm the Programme Director from University College London, and I'm delighted to be chairing today's webinar. This is the first of a series of 5 webinars, which will take place on the last Friday of each month. In each webinar, we will have presentations from an Alzheimer's Society-funded researcher who will be presenting on their work, and a different careers talk each time, which will help you hopefully both practically and to expand your understanding of some of the great work being funded by the Alzheimer's Society as well. I'm delighted to introduce today's speakers. Firstly, we will be hearing from Coco Newton. She is a PhD student from the University of Cambridge. Coco is exploring spatial navigation and cognition in early Alzheimer's disease, and she's previously worked on the C-Hero Quest Citizen Science Project and is now taking this further with the use of virtual reality. So very much looking forward. Coco, would you like to turn your camera and microphone on and just say hello?
Hi, thanks for the introduction.
Thank you, Coco. Our next speaker is Dr. Claire Jonas, who is a research communications officer from the Alzheimer's Society, a former rehabilitated academic who worked in psychology and neuroscience previously, and she has also very smartly turned her hand to public engagement and supporting early career researcher careers. Hello, Claire.
Hello. Thank you for calling me rehabilitated. That is a very good description.
Each of our talks will take up the 90 minutes that we have in total today, and we'll have time for questions after each of the sessions. So please do remember to leave your questions, and thank you very much, everybody, for joining us. And Coco, perhaps if I can, I'm going to turn my camera and microphone off now and invite you to deliver your talk. Thank you.
Thank you very much to the organisers for inviting me to give the talk today. I'm really happy to be chatting about my work that I'm doing during my PhD. I'm just entering my 4th year, and because of COVID, there was a lot of delays to data collection, so I only have very preliminary results to show you, but hopefully it gives you a taste of what's to come. So my PhD study is called the ENCRYPT study, and I will be referring to that throughout the talk. And ENCRYPT stands for Entorhinal Cortex Hippocampal Circuit in PREVENT. PREVENT Dementia is the cohort that I'm working with. But a much more interesting title is Virtual Reality Navigation Tests. Could they be new cognitive markers for preclinical Alzheimer's disease? And I'm in the Department of Psychiatry at the University of Cambridge, and I'm being supervised by Professor John O'Brien and Professor Li Su. So why do we need new cognitive tests in Alzheimer's disease? Do we need to improve them? So if you look at this diagram of the entire continuum of Alzheimer's disease, so imagine time is moving along the x-axis here. You have individuals that will progress along the green line through life. They will never really accumulate many Alzheimer's biomarkers or pathologies such as amyloid and tau, the 2 hallmark proteins, and they'll live till the ripe old age of 100, completely biomarker free and free of cognitive impairment. You might have some individuals that progress along the dark green line through life. So they start to accumulate amyloid and tau, but they also live to the ripe old age of 100, and they will never really experience severe cognitive impairment. But there will be some people on the dark green line that start to follow up the dotted green line and eventually cross the threshold to be diagnosed with clinical Alzheimer's disease dementia. And of course, this whole process and why people start to go up the dotted line is very unclear to us. It's likely a multifactorial etiology. We know that there's lots of factors that influence people's risk for going up that line. So there might be your age, your genetics, if you have comorbid diseases, how much cognitive reserve you have. And we know that there's lots of factors that indicate that individuals are potentially going along this line. So that might be cognitive changes, it might be structural or functional connectivity changes in the brain measured by MRI. It could also be amyloid or PET changes on a PET scan, where you have a radioactive ligand injected and you have a brain scan, or it might be cerebrospinal fluid sample or a lumbar puncture sampling of those proteins. But out of all of those indicating factors that we have, cognitive changes are the most straightforward, simple, cost-effective, least invasive way of measuring if someone is about to go up this dotted line. The only problem is that current cognitive tests are designed to detect people at this point, at the point at which you're starting to get dementia. So the Adam-Brooks cognitive exam, the mini-state mental, they're all designed to detect dementia. But ideally, what we want is some sort of cognitive test that is capable of picking up people that are just about to start progressing along this dotted line here. And we don't yet have those cognitive tests. Another reason that we need to start improving what we're using in Alzheimer's disease in terms of cognitive tests is an issue of translation. So in the clinical trial continuum, when a new drug comes through from the development guys at the pharmaceutical companies, they'll give it to an animal model of Alzheimer's disease, and then the animal model has to swim in a milky pool of water quite often. And the faster they find a hidden underwater platform, the better the drug is having an effect at slowing down the Alzheimer's progression in their brains. And if the drug passes this stage, then we go to humans, and then humans get to do this complex visual patterning reasoning question. And for some reason, it's not making humans better at doing complex visual pattern reasoning, but it was good at making animals better at spatial memory. So one thing that we could be doing is actually being more valid in terms of how we're evaluating the effectiveness of a drug in changing a certain behavior. So if we take a theory-driven approach to how we could approach designing new cognitive tests in Alzheimer's disease, we could say, well, we know that the tau pathology is the pathology in Alzheimer's disease that correlates best to the severity of cognitive impairment. And that's measured quite nicely by these Braak stages, which have been around for decades. And we know that the tau pathology starts in this select region here, the entorhinal hippocampal region. And that's a region made up of lots of different subregions. And so the question is, what functions does this region do that we could test? Well, we know that the hippocampus and the entorhinal cortex does episodic memory, but something that's lesser known, and also why it's tested in animal models, is spatial memory. And that is a really important function of the entorhinal hippocampal circuit. And that is because it harbors very unique types of cells that are responsible for our ability to navigate, and they're sort of quasi GPS cells of our brain. So if you imagine we need to navigate, step one, you need to know where you're facing. So we have head direction cells that fire when your head is facing a certain orientation. Step two of navigation, you need to know where you are. So we so happen to have place cells which fire at specific locations in environments. So these were discovered by recording from a rat while it was foraging around a box. And at the same time, the location of the rat was being tracked. And they found that when rats crossed specific places, known as the place field, one cell would just fire. And that was thought to provide a mechanism for the rat to be able to self-locate inside this environment. Grid cells are similar to place cells. They provide self-localization information, but instead of one firing field, they have multiple, and they're arranged in this really nice hexagonal array. And it's thought the function of grid cells is to provide a sort of metric of space. So step three of navigation is being able to know where you're going. So if you can imagine this rat's traversing all of these fields here as it walks along, and it knows what distance and what direction it's traveling in. And then not only that, but you need cells to be encode what's immediately around you in your environment. So it, again, just so happens we have these boundary vector cells, which have receptor fields that are anchored to boundaries at specific distances and directions from us. So they cover all four or all eight or however many boundaries there are in your environment. And we also have object vector cells, which have receptor fields tuned not to boundaries, but instead to select objects in your immediate environment. And altogether, it seems like these cells provide a sort of array around us that are signaling what's near us, where we need to go, how to get there, and things like that. But the important thing is that these spatial cells are present not only in rats where they were first discovered, but also in humans, in bats, in primates, in birds. So what we're looking at here is a very evolutionary conserved mechanism present in multiple species. It happens to be the behavior that we test in preclinical animal models. So we're ticking the translation box. But also navigation is a very ecological language invariant behavior that every human has to do every day. So as we move towards more global clinical trials, more global research studies, it's important to start asking how can we do cognitive tests where we don't have to translate it into hundreds of different languages. So navigation is a really great candidate. But the most important reason why navigation is such a great candidate is because these cells are so specific to the regions that are very heavily affected by the earlier stages of Alzheimer's disease pathology. So if you look at the dark green arrows here, we can see the progression of tau pathology. We know it starts in the entorhinal regions. It then moves into the hippocampus, and then it moves posteriorly to the posterior cingulate cortex. And these are yellow regions which are affected in the preclinical stages of Alzheimer's disease. We know that amyloid starts diffusely in the cortex and it moves downwards into the hippocampus progressively. And when we look at these spatial cells that I've just talked to you about, what's very interesting is that they reside within very unique locations within this pathway. So grid cells and boundary and object cells you can find in the entorhinal cortex. Place cells you can find in the hippocampus. And head direction cells you can find in posterior cingulate and retrosplenial regions. So what if we had tests that could target the function of each of these cellular populations? Now, of course, navigation is a network dependent behavior. All of these cells interact all together all the time to enable us to navigate, but they do have special functions that they do by themselves that we could potentially target with spatial tests. And we've already begun to look into that with the virtual reality supermarket test, which is very innovatively named, but it's a very effective test, and the Four Mountains test. And these two tests are specific to different regions: retrosplenial cortex for the virtual reality supermarket test, and the Four Mountains test is specific to hippocampus. So when we look at how these do in different populations of Alzheimer's disease, they're really effective in the more symptomatic stages of the disease. So in Alzheimer's disease, we know that atrophy of the posterior regions here is very specific to Alzheimer's disease. You don't get it so much in other types of dementia like frontotemporal dementia or semantic dementia. So when you give the test targeting the region, the function of that region, we see a really nice dissociation where in white, Alzheimer's disease patients are really awful at it, but other patients such as semantic dementia or behavioral variant frontotemporal dementia patients are bad at it. And this task is essentially watching a triangle move through a supermarket, and you turn around a couple of times and you just have to point in the room back to where you thought you started. And this is just presented on an iPad. So it seems to be that if you do target regions specifically affected, or the spatial function of regions specifically affected in Alzheimer's disease, you might pick up on something that's more sensitive and specific to Alzheimer's. And even in more prodromal stages, the Four Mountains test is showing some evidence of being good at delineating the different types of dementia. So in a cohort of healthy controls, prodromal Alzheimer's disease patients, so that's MCI or mild cognitive impairment, you have two populations, one that are negative for Alzheimer's disease biomarkers and ones that are positive for Alzheimer's disease biomarkers. And then you have the Alzheimer's disease patients themselves. And you can see this really nice linear trend where the severity of Alzheimer's disease pathology is related to a much poorer performance on the Four Mountains test. But of course, the goal of the ENCRYPT study and this talk is about preclinical ID. So we know that there's some sort of signal happening in later stages of the disease. What about the first stage of the disease in this entorhinal region here? Could we possibly design a test that targets the function of these cells in the entorhinal cortex? And that's where these new tests come in that I'll be talking to you today in the ENCRYPT study. So the ENCRYPT study is using a whole battery of virtual reality spatial tests that are sensitive to different points in the Alzheimer's disease tau pathway. And in combination with these different virtual reality tests, I'm also using 7 Tesla MRI. Now, 7 Tesla is about double the strength of the usual MRI that you get in standard NHS clinical scans, which is usually about 3 Tesla, sometimes 1.5. And the reason that we're using this high resolution imaging is that we can delineate the regions of interest in the entorhinal hippocampal circuit with extreme precision. So in brown here, for example, you can see the entorhinal cortex, then in here, the subiculum, CA1, CA2, CA3, different subregions of the hippocampus. And the goal is to ask, does different levels of Alzheimer's risk in the prevent dementia cohort, so that's a cohort of individuals aged between 40 and 65 years, does their performance on these tests correlate to their risk of Alzheimer's disease in midlife? What measures of Alzheimer's disease risk are we using in midlife? Well, we have a whole selection, because the wealth of the prevent dementia cohort, which is a nationwide cohort with different sites in Oxford, Cambridge, London, Edinburgh, Dublin, individuals every 2 years are assessed for lots of different things. They have blood tests, lifestyle questionnaires, physical exams with doctors, and they also undergo a very rigorous full day of cognitive testing. But we know what the ApoE4 status is. So if you have the ApoE4 allele, you're 2 to 3-fold more likely to develop late onset Alzheimer's disease. We also have measures of polygenic risk, because we're starting to understand that Alzheimer's disease is probably not going to be linked to one gene, but many, many genes that have very small effects together. We also have biomarkers, so cerebrospinal fluid or PET biomarkers. We also have things like dementia risk scores. So the CADE dementia risk score is related to cardiovascular health factors. So they include scores for how good is your blood pressure? Are you physically active? What is your BMI? what is your age, what is your gender, and you get a score out of 16, with the higher score meaning a worse cardiovascular health, which we know is also related to risk of dementia. And we also have family history status, because parental family history is also related to a higher risk of Alzheimer's disease. Of course, we don't know for sure that these participants will get Alzheimer's disease, but we have these compiled risk factors. We can begin to ask questions, and the goal is over time to follow up this cohort until they do or do not get dementia, and retrospectively look back at what was the best sign that we could use to predict who will get dementia. So these entorhinal tests then, what would we use to target the function of the grid cells in the entorhinal cortex? So in the entorhinal cortex, we know that the earliest changes in cerebral blood flow are around the lateral and the medial entorhinal cortex. The special thing about the medial entorhinal cortex is those grid cells that I mentioned to you at the beginning of the talk. So as I said, we have a rat that's being recorded from the hippocampus in the entorhinal cortex. It's foraging around a box while its position is being tracked. And you start to get these maps of these firing rate maps of different cells. So when you put lots of different electrodes together, you can compile what's known as a spatial autocorrellogram. But basically what it's saying is, here's a grid cell and here's all of the firing fields. And the grid score is 0.8 out of 1, let's say. So it's a really nice grid cell. It's got very nice symmetry, very stable. When you do the same thing in terms of recording from a grid cell in a rat that's been transfected with a human tau gene, what you see is that the grid score completely drops, and you don't have the nice regular symmetry. They're very unstable grid fields. So it seems like Alzheimer's disease is having a really significant effect on the cells. So the question now is, what's the behavior that's dependent on these type of spatial cells? And the answer to that, we think, is something called path integration. Path integration is the ability to know where you've been in space. And it's very nicely exemplified by ants. So here is a diagram of an ant leaving its nest. It travels hundreds and hundreds of meters, despite being the tiny little thing that an ant is, turns around hundreds of times, but when it finds food, it's able to just beeline straight back to its nest. It has no trouble of calculating that vector. So the whole time in blue, it's been integrating where it's been going and in what direction it's been going. So that path integration, we think, is dependent on grid cells, because remember I said that we think that having a repeating fields enables us to have a metric in our brain of how far and what direction we're traveling in. So can we test this path integration behavior in humans? And the answer now, with the advent of immersive virtual reality, is yes, because we can put people inside a virtual world where they have their full body motion available, their full idiothetic vestibular cues, their proprioceptive cues, in which they're able to estimate where they've been in space. So they'll be in an environment like this one, and the task is very simple. They just start at a random position, and they have to walk to cone number 1, and then cone number 2, and then cone number 3, at which point the cones have disappeared. And the goal is just to walk back to where they think they found cone number 1. And let's say they happen to do the trajectory in red instead of the real trajectory in blue. We can measure the distance error, we can measure the angular error, and lots of other things. And then the beauty of it, because it's virtual reality, is we can do things to the environment to make it even harder. So remember that I'm going to be testing people who are middle-aged, seemingly completely healthy. So we really need to stress out their entorhinal cortex and their path integration ability. So what we can do is take away information from the environment. So for example, here you can see that there's mountains, which maybe in the north of the environment there's mountains, and then in this example, they're gone. So they have no orientation information. And the PhD student before me, David Howitt, he took this virtual reality path integration task and he gave it to a cohort of mild cognitive impaired individuals. So that's that prodromal stage of Alzheimer's disease I was talking about. And the individuals with biomarkers of Alzheimer's disease are more likely to progress to Alzheimer's. The individuals without the biomarkers are less likely to progress to Alzheimer's. They're more likely to either have some form of stable MCI or progress to a different type of dementia. And what we can see is that there was a really nice delineation between individuals that were at risk of Alzheimer's disease versus not. And even more impressive was that when you compared this test to current gold standard cognitive tasks that we use in the clinic, so the free and cued selective, the ACE, even the 4 mountains task, which was that hippocampal task I was talking about, it has superior classification accuracy. Now, these results, of course, they need to be replicated and we need to do it in larger sample sizes, but there is some suggestion that there's a signal here. And the question now is, how early can we pick up on signs of incipient disease processes in individuals? So I've been testing 83 people so far in the PREVENT cohort. So these are middle-aged individuals aged between 40 and 65, and they've done this virtual reality task. So the first thing that we'd wanted to see is, are we stressing people's entorhinal cortex out? Are we stressing out their path integration ability? So in the virtual reality task, there are 3 conditions. We have the no change condition, where participants have all of the spatial cues available to them. We have the no optic flow condition, which is we basically take away the ground. So participants, as they're walking, there's no optic flow information. They don't know how fast they're walking. We think that's possibly going to stress their ability to estimate distance. And finally, we have a no distal cues condition, which is the one I already mentioned, where we take away the horizon, so there's no orientation information. And what we can see is that as the task gets progressively harder, we do see that there's a significant increase in distance error on the y-axis. And now the question is, how does your risk for Alzheimer's disease affect your performance on the task? And somewhat unsurprisingly, I think, we don't see any group differences on principal risk factors such as family history at the top. So in blue, we have people without a family history, and in yellow, we have people with a family history. There's a slight trend that people with family history perform slightly worse on the task, but it's not significant. And there's absolutely no rhyme or reason to whether or not you have an ApoE4 allele. And these are the only 2 risk factors that I've had time to analyze so far. But I think, you know, for middle-aged individuals, it's not surprising that we're not seeing any performance deficits. However, what if we dig into the data a little bit deeper? So when you take family history de facto, it's also important to look at paternal versus maternal family history, because we know that there are gender-related inheritance in Alzheimer's disease. So what an interesting finding so far has been is that if you take males with a paternal family history in yellow, they perform significantly worse than males without a paternal history. If you take females with a maternal family history here in yellow, they also perform significantly worse than females without a maternal history, but you don't see the same effects in the opposite genders with the opposite family history. So there's something to follow up. Still very early days. I don't know quite what it means. I don't have a hypothesis yet as to why that might be. Maybe there's some risk genes associated with the X and Y chromosomes. If anyone in the audience has any suggestions or has ever seen an effect like that before, please do say something. And the other interesting thing we've been finding is that ApoE4 carriers seemed to be very effective by the distance at which they have to walk. So on the x-axis is the length of the incoming distance of the triangle. So when they're walking from cone 3 back to cone 1, the longer that distance is, the harder or the worse the ApoE4 carriers do. But we don't see the same effect in people that don't have an ApoE4 allele. So there seems to be some sort of unmasking of the deficit when you increase what you're asking the path integration of these ApoE4 carriers to do. So a very early day still, and lots more to come. Hopefully in about a year or so, I'll have the full set of results. But now I'm going to leave the path integration task and switch over to the neuroimaging side of the ENCRYPT study. So if you don't know anything about neuroimaging, that's fine. I'll take you through it. When we take an image of the brain, we do it in slices. So basically from the front to the back, a slice, 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 slice. And they tend to be at 3 Tesla, the sort of standard imaging strength. They tend to be around 2 millimeters thick. But if we just, if you look at this as the entorhinal cortex, so it's just the cortical bit of the entorhinal cortex at the base of the brain here. The medial entorhinal cortex is this portion in brown, and in standard clinical imaging, you can just get it in about 2 slices. So you don't have much data. Despite that, David, the PhD student before me who took this virtual reality path integration task in the cohort of MCI patients, he found that there was a relationship between how big your entorhinal cortex was and how well you did in a task. So the higher your distance error, the smaller your medial entorhinal cortex. But in the mild cognitive impairment stages, you are likely to see quite overt structural changes, quite overt atrophy in these patients. But of course, the PREVENT cohort are middle-aged. So we need to have a slightly stronger magnet in the scanner to get slightly higher resolution images so we can look at this medial portion of the entorhinal cortex in greater detail. So if you are interested in the protocol of the MRI sequences that I'm using, I've got them here. But essentially what we're doing is we're saying we don't care about the rest of the brain. We just care about this very small portion here. The blue is the anterior lateral entorhinal cortex, and the yellow is the medial or the posterior medial portion. So we're just going to take very, 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 very thin slices of the brain in the region we care about. and look at it with exquisite detail. So these are the images that we can get out with 7 Tesla. And what we're able to do then is examine the medial portion of the entorhinal cortex with about 10 slices instead of 2 slices. So this is the whole entorhinal cortex, which looks a lot more precise than this blocky one over here from 3 Tesla. And then we can split it into the medial and the lateral portions here like this. So we have a much more accurate estimation of people's medial entorhinal cortex. So the next stage is to take these measures of the entorhinal cortex and relate it back to that virtual reality performance. Do people with a smaller entorhinal cortex make a larger distance error, and is that related to Alzheimer's risk? The final aspect to the study that I wanted to talk to you about, if I have time, I think there's still time, is what about looking at the function of the grid cells themselves? So it sounds a little bit wacky, but bear with me. If you look at this image of a grid cell here, we've got the nice hexagonal array of firing fields. There might be a way to look at the firing of these grid cells using fMRI. So fMRI is functional magnetic resonance imaging. Instead of looking at the structure of the brain, we're looking at the function of the brain, or a proxy measure of the function of the brain by looking at changes in blood flows in regions that we're interested in. And we correlate these changes in blood flows to events that someone's doing in the scanner, to a task in a video, to see if we can predict when the signal is going to change. So why can we look at grid cells in fMRI? Well, you know that there's the hexagonal 6-fold pattern of firing like this. And the other thing to know is that when you walk in alignment with your grid, so from this cell to this cell, the firing of this grid cell will increase. Whereas if you walk in not aligned with the grid, so from this cell in between these 2 cells, the firing rate won't change. So if you take one grid cell, you'd expect the firing to go up along these paths, these movement directions in an environment. And then, of course, we don't only have one grid cell in the entorhinal cortex, we have a whole population of grid cells. So we should be able to see a macroscopic signal increase when people are walking in an environment that's aligned with their grid. So we take people doing a task, we estimate where their grid events are going to be, we iteratively estimate the orientation of a grid in one half of the data, and then we test that orientation in the second half of the data. And all of the previous studies that have done this type of experiment here, so there's quite a few of them, have always found that this kind of increasing signal every 60 degrees is often found in the right entorhinal cortex. So in a pilot study that I did a couple of years ago, which I'm now running in PREVENT, we had this object location memory task, which was basically asking people to walk around a room and they just have to remember where different random objects are. But actually, we weren't interested in how well they remembered the objects. We were interested in finding out whether they were mapping this room and getting their grid cells to path integrate as they were walking around. And what we found in the pilot is that, yes, we did see the strongest increase in the right posterior medial or medial entorhinal cortex. And we did that, we ran that against a lot of control analyses to show it was specific to 6-fold symmetry. So we also tested, okay, what if we had grid cells with 4-fold symmetry or 5-fold symmetry or 7-fold symmetry? But it really was the 6-fold symmetry model that was working. So the next step is to take this type of analysis, this task, and see if in the PREVENT individuals that are doing the behavioral path integration task, which is dependent on these grid cells, how is their behavior, how is their grid cell functioning in fMRI, and what structurally is the entorhinal cortex like? Are there any entorhinal changes? So we're sort of interrogating the entorhinal cortex of these preclinical, possibly preclinical Alzheimer's disease patients from every way that we can think. So to summarize everything that I've just been through, we need to create new cognitive tests that are sensitive to preclinical AD, and that's an indisputable fact. There's lots of different ways to go about it. But the theory that we're taking is, well, we know that the tau pathology, which is the pathology strongest correlating to the clinical profile, starts in this region here, the entorhinal region. And we know that that entorhinal region uniquely houses cells that are dependent on different aspects of navigation. So we know that spatial navigation tests designed based on cellular understanding are specific to the earliest AD regions. We think that they could possibly be capable of actually tracking pathology progression. So you can start to see how different levels of impairment occur as the pathology is potentially moving through, although that still needs to be examined. But also, importantly, the navigation tests are ecological and completely translational with what we're doing in preclinical animal models. And that's made possible through virtual reality. So this image here is a rat running on a styrofoam ball. it's completely surrounded by a virtual task. And here is one of my participants wearing a head-mounted display, and they could, in theory, be doing the exact same task. So that's how translational we're now able to get with virtual reality. So the preliminary results that I have with the PREVENT cohort suggests that potentially some AD risk factors are associated with worse performance if you dig deep into the data, but that still needs clarification, and there's lots of other risk factors to look at. And ideally what we would do is create a whole model with every single risk factor we can think of to see how people perform on those entorhinal tests. And the next steps are to compare this performance on the behavioral path integration task with the imaging data that we're getting from the super high resolution 7 Tesla images. But also more importantly is to look, how do these participants in the PREVENT cohort do on current gold standard cognitive tests? Because if they do badly on the entorhinal cortex test, but they also do badly on other cognitive tests, then it's kind of suggesting that we're not really achieving anything with this theory we've got here. But that remains to be seen, and hopefully you can log into the next talk I give where I can give some more updated results. So thank you very much to my supervisors and my collaborators and my funders, and thank you very much for listening and joining the webinar today.
Thank you, Coco. That was fantastic. Thank you so much. And we've already got questions for you as well. And I should add that as a participant in the PREVENT study,
Really?
I am. I'm older than I look. I'm looking forward to maybe getting a chance. I live in Oxford, but I participated in London, so maybe does each site perhaps have different studies running on their cohorts?
Well, we sent out a recruitment email to the West London cohort a couple of months ago, so maybe it just slipped by you. But if you're still interested to take part, we're still recruiting.
Definitely. Anything with virtual reality, I'm kind of love that. If you can double it up for some games as well, I'm all of. Thank you very much. That was really fascinating, and I'm particularly interested to know how you managed to map the movement of ants. but that's just...
that wasn't me. That was someone else. I didn't have to go through that torture.
I have this vision of little tiny trackers being attached.
Yeah.
Okay, we've got questions. Claire Jonas, our other speaker for today, is going to get her question first. I'm just going to remind everybody who's watching on Zoom or YouTube, please remember to post your questions, ideally using the Q&A button rather than the chat if you can. But don't worry if you've already done that. I'll be sure to pick those up. So the first question is to ask, what other cognition tests you think might be used alongside this one to predict clearly? I mean, this is, we had a presentation earlier in the year discussing gait analysis and other different forms. There's quite a few of these cognition kind of tests and biomarkers coming out. Do you have any thoughts on that?
Yeah, I mean, absolutely. I don't think there's going to be a one-shot wonder for one cognitive test at this stage. I see it as a whole battery of different tests that are specific in some way to the earlier stages of the disease process, which might be tau, but it might be other things. Yeah, I think gait is a really good way to go. There's other studies looking at, because we know that the entorhinal cortex and the hippocampus also does episodic memory, but instead of thinking in terms of word list learning and list recall, what about the content of episodic memories? There's a lot of research looking at the fact that the more posterior regions of the hippocampus tend to be focused on the context of a memory, whereas more anterior regions tend to be focused on more sort of the content, the objects, the people of the memory. So if you start to probe those individual functions, you're also getting down into very specific functions of the regions that are subserving episodic memory. So I think there's a lot of exciting research going on, but I think the key will be specificity of regional function rather than looking at global cognitive tests. That said, also, there's a lot of research looking at composite cognitive markers, so taking lots of different regions and putting them together and look at how people are doing globally on those little individual regions as opposed to just one region at a time. And I think that's also going to have a lot of sensitivity.
Yeah, that makes sense. I mean, and we did a podcast on this a few weeks ago talking about the importance of diagnosis and early diagnosis and what you're going to do with that. And I suppose it's that knowing this earlier, of course, does provide a great then cohort of people to test out new treatments on, which of course we struggle with at the moment. And because giving the right drugs to the right people seems to be the key if we're talking about drug therapies. Or of course, trying, as new treatments come through, we're going to need to target those at the right people. Yeah,
it's an important point you raised that I didn't really have time to touch on in my talk. But when we talk about cognitive tests, we're not only talking about ways to detect incipient Alzheimer's disease, but how to monitor it. And if you're going to start putting patients at earlier stages through clinical trials, how are you going to evaluate how well a drug or a therapy or a prevention method is doing if you're not targeting the functions of the right regions?
Absolutely. And being able to discount other potential, being able to single down to say this is Alzheimer's disease or this is FTD as opposed to something else that's going on. We have a question from Sarah Pape, I hope I've pronounced that correctly, who asks, do you think grid cell involvement could be related to the early difficulties people with Down syndrome seem to experience in Alzheimer's disease? Lots of people we work with have depth perception difficulties, which I generally assume to be vestibular related, but I wonder if it could also be related to navigational cells.
Yeah, I'm not too familiar with the literature around Down syndrome, although I know there is a very strong overlap with Alzheimer's disease. I wouldn't be surprised if they were involved, purely because there's a lot of theories as to what drives the firing of grid cells, but one of the strong theories is it's actually cues from the vestibular system and cues from the visual system and cues from coming up from the thalamus that receives body feedback from muscles and the receptive cells we have in our muscles. And it's thought that those possibly are kind of integrated into estimations of distance and speed and the head direction cells provide that orientation information. And the role of the grid cells is to take all of this information together and give you a kind of holistic idea of where you're traveling. But if one of the sources of those information starts to break down, or the grid cells themselves, then you might start to see breakdowns in different aspects of your ability to navigate. And that might well be related to depth perception, for example. But I don't know. It's a really good question, and it would be good to read up on that.
And we've talked before, not obviously as part of this series, but as part of the podcast in trying to encourage people to think about dementia in careers. I think your work really well kind of demonstrates how people from all different backgrounds can bring their talents and interests and apply those to dementia. I mean, I assume in devising these tests that you're using, are you partnering with people who are writing the coding and doing the modeling for
Absolutely.
the software that runs on the?
Yeah, absolutely. So I work with the guy who coded the path integration task. He was a PhD student at UCL at the time. He's now a postdoc, but he's super interested in game design, and he'd love to go into the game design industry because he had just had so much fun building this whole virtual world. And I work with another project I did at UCL, also on spatial navigation. I had to work with engineers to build this whole real world replica of the virtual test I was doing. I need to work with all sorts of modelers, virtual reality experts. It's a really exciting field to be in, purely because you have your fingers in so many pies and you never know where the research is going to take you.
Yeah, I think I might have met those. Are they the same team that are looking at eye tracking and things like that as well?
Could be. They built a whole model of the London Tube system to try and work out how people move in big crowds to get on and off the train, and how they could optimize that with different lighting cues or different sound cues. It was super interesting.
And the technology allows this to be done safely as well. It's not like it's attacking an ankle bracelet somebody and say, right, go for a walk and let's see if you can drop somebody in a town and say, find your way home.
Exactly. Although that's definitely the beauty of virtual reality, is that you've just got so much control over what people are seeing.
And tying in, I think, obviously the big tech firms are in on this as well, aren't they? I know I've seen YouTube videos from Google and Amazon and others that are working in this space as well. Fantastic, fascinating. If there are no more questions for Coco, last chance, and I'm just going to remind you that our next talk, while you're having a think about that, comes from Claire Jonas, who's going to move on to our careers talk session of today, and is going to talk about academic stresses, something that I hope you don't have a problem with, Coco, but I know many people do. So thank you very much. We're getting lots of comments back saying great talk, and I completely agree. Thank you so much, Coco. And Claire, if I can invite you to turn your camera and microphone on, and if you'd like to share your screen.
Hello everyone, and especially hello to my old office mate Stephen Jeffs. I didn't expect to see you here, but you have seen me go through this firsthand, so hopefully it won't be boring for you. So I am an expert in the mind and brain, I imagine, like a lot of people who are here. So I've got an MSc in neuroscience and a PhD in psychology, and I'm going to be talking about mental health stuff today, but I just want to be clear, I'm not a trained therapist, I'm not a counsellor. I'm talking about this largely in terms of having reviewed the research that other people have done and my experience of having been through therapy myself for pretty bad anxiety. I've also been a PhD student, a teaching fellow, a postdoc, and a lecturer, and then I burned out. So I've experienced what academia is like from the inside, and I really know how rough it can be. So today I'm going to focus mainly on the research on stressful waiting and mental health, because a lot of the stresses in academia are about waiting. Occasionally I'm going to talk about my own experience of things that have helped me and move away from the research, but I'll let you know if I'm doing that. Anything that is research based has a citation on the screen, and you can ask me for a full list of them afterwards if you want. Anything that's personal to me, I will signal that. There is also, if you want it, a full reference list available and a summary sheet of what I'm going to talk about in case you're sort of frantically scribbling notes. So if you want either of those things, just email me or find me on Twitter. I am thatthinkfeel, and you can tell me if you want either of those, I'll send them to you. All right. So first question we might want to ask is, what kind of stresses do academics face? And I am going to talk about stressful waiting today, but there are also other enormous systemic stresses that you're going to be facing. Academia is super weird, and I didn't really realize how weird it was until I left. One thing that I found particularly weird was the insistence I heard from people inside academia and outside academia about how lucky I was to be doing a job with such variety and freedom. And I mean, yeah, that's true. And I did feel like that some days, but it also, talking about it in that way, disguised the fact that there were big problems. So very briefly, inequalities, because historically academic spaces were restricted to certain kinds of groups. So white, male, rich, either didn't have kids or didn't have to engage in much childcare. And those kinds of restrictions are nominally gone, but they hang around under the surface, and it can make academia harder if you don't belong to those groups. There's also short-term contracts and job insecurity. So at the early career stage, there's an expectation that you'll uproot and move to a new university, a new research group every few years, regardless of whether that's what you personally want to do, or if it's possible for you, even given other commitments that you have in your life. And not knowing whether you're going to have a job in a few years or even in 6 months can take a real toll. This comes alongside silence around mental health and illness. So both of those things that I've just mentioned are enormous stresses, and it's an intensely pressured environment. But talking about the mental health consequences of that can be met with disdain or hostility. And this is one of the things I really hope has got better since I left academia. And lastly, quite often you're encouraged to have poor boundaries between your job role and your other aspects of identity. So a couple of good examples of this. One is about taking time off. Even a weekend can be viewed with sort of some suspicion. So one academic I know, she posted an article on Facebook recently that was encouraging academics to switch their email off just for one day. And I expressed my concern that never switching off from work was so common among academics. And her response was, oh, that's just part of being a professional. I don't work a 9-to-5 office job. And I thought about this and I went, I'm also a professional. I can tell you that constantly being on for your work is not a requirement in professional jobs outside of academia. And honestly, it wasn't a requirement inside academia either, despite what you might be hearing from other people. So my way of dealing with this now is, will something actually catch on fire if I don't think about this right now, or if I don't read to this email right now, or if I don't respond to this email right now? And if the answer is no, doesn't need doing. So related to that, you're also often expected to have no boundary between the self you are at work and the other aspects of your identity. So one other researcher I know once described himself to me by appending scientist to every other description. So he was a scientist father and a scientist spouse. He wasn't a scientist, a father, and a spouse. Being a scientist came into every aspect of his identity. And that kind of blurring between your work self and your other selves can make things really difficult if work isn't going well. So, as I said, these are problems that you really have very little power to change as an early career researcher. And I'm going to talk about actions that you as an individual can take to alleviate stress, but I want to be clear that the stress itself is often coming from the system, not from you. I'm also going to be talking, as I said, mainly about stress related to waiting, but this is advice that's applicable to other kinds of stress as well. Like, all of the stuff that I will talk about is good for any kind of stress you're experiencing. It's just I'm framing it as waiting related stress. Okay, so academia has a lot of stressful weights in it. Will this experiment work? Will my talk go well? How's this interview going to go? Will my paper be accepted? Will I get the grant I've applied for? And I've arranged these in approximate order of waiting time, although obviously some people's experiments take much longer than others. So move that one as appropriate. But you can be dealing with stressful waits for, you know, minutes to months to the point of the COVID pandemic starting and maybe not being able to collect data anymore, potentially years. We didn't necessarily know how long that was going to be, or even if it's going to come back at this point, which is very exciting. So with that in mind, I'm going to talk about the kinds of reactions that people have to stressful waits. So if you're a psychologist or you're working in a similar field, you'll probably be familiar with the WEIRD problem, which is an acronym. So Western, educated, industrialized, rich, and politically speaking, democratic participants are the people who tend to take part in research. So largely university students in North America and Europe. We don't have as much information about people who do not belong to those groups, and generalizing from the WEIRD population to other people doesn't necessarily apply. So just bear that in mind when I'm talking about this. A lot of the research has been done on the WEIRD population, and specifically on, in the case of these, law students at university in California who were waiting for the results of their bar exam, which for some reason takes 6 months. So my students would have been very mad if I'd taken that long to return their essays to them, but okay. I guess law is very different. So keep that in mind. All right, people react in different ways to stressful waits, but some common reactions that we know about are a drop in the 3 fundamental psychological needs. So these are competence, which is how good you feel you're doing at your job or in your life in general. Connectedness, how well you feel connected to other people. And autonomy, which is how well you feel your behaviors reflect your values. All of those can tend to drop. Quite often, competence will drop in a weird U-shape. So if you've gone through an intensely challenging thing like writing a grant application, you will probably feel less competent immediately after it, and then it'll rise. Then it will drop again around the time that you're expecting the grant application results. Secondly, you might get sleep disturbances, so insomnia, nightmares, restless legs, whatever kinds of sleep disturbances you experience. Difficulty maintaining positive health behaviors. So these are things like getting exercise, eating a varied diet, that kind of thing. Because you're stressed, a lot of your attention can go on dealing with your stress, and it can be difficult for you to put your attention on the positive health behaviors, which may also require some effort to keep going. Unsurprisingly, you may spend more time worrying than usual as well. So not everyone is going to experience all of these, not even any of these. Some people deal really well with stressful waits and some people do not. So you might be going, well, why is that? The answer is basically there are individual differences in how much we worry depending on 5 different characteristics of the weight and of ourselves. So firstly, how long till the wait is over? How much control we have over the outcome. Weirdly, a medium amount of control seems to be good. Low levels and high levels of control are not great. Perceived risk of a bad outcome. So this is definitely perceived risk, not actual risk. Perceived severity of a potential bad outcome, and some individual differences, which include gender, pre-existing mental health problems, and your sort of inherent levels of optimism. So I'm just going to work through this briefly in terms of 2 people who are waiting to hear back about a grant application that they've jointly made. So person A is worrying much less than person B. So there's no difference between person A and person B in terms of how long until the wait is over and the level of control that they have over the outcome. Both of those are going to be the same. But the perceived risk of a bad outcome could be very different. So for example, person A might think that the grant application is pretty likely to succeed and they have therefore a low perceived risk of a bad outcome. Person B thinks it's probably going to fail, high perceived risk of bad outcome. We're also potentially going to see differences on the perceived severity. So for example, person A is on a permanent contract. They are not going to experience particularly severe consequences if they don't get this grant. Person B is on this on a temporary contract and is relying on this grant to stay in academia. And lastly, those individual differences. So let's say in this case that person A is male and person B is female. Men generally report less worry about stressful waiting than people of other genders, but it's not clear if that's a genuine difference in how much different genders worry, or if it's about the social expectations that different genders have placed on them. And there are also other individual differences that are linked to worry levels, like how well you generally cope with uncertainty, whether you're a mindful person. So all of those can feed into how much you're struggling with a given stressful wait. All right, let's talk coping tactics. So none of these are guaranteed to work for any one person. I do not want you to think of this as a recipe for getting yourself out of a stressful mental space, but rather a menu of things that you can try in order to get rid of stress. So first of all, the very obvious, you're dementia researchers, so you probably already know the mantra, what's good for your heart is good for your head. And that's both physically what is good for your brain and mentally what is good for your mind. So obviously you want to be trying as best you can to do things like eating a varied diet, drinking enough water, getting enough sleep, and doing some physical activity that you like. So here you have a picture of a big hill, because it turned out when I moved here from London a few years ago that big hills in particular were very good for my mental health. But for you, going into a different environment might be good. Particularly here, I've put up a natural environment. So something like a forest, a park, big hill, rather than an urban one like a street, is particularly helpful for things like lowering struggling with my words there, lowering blood pressure and lowering stress hormones. Obviously, you might not be able to access natural spaces particularly easily. So if you have a disability that restricts you to the house, or if you simply don't live in an area with many parks, you can take virtual walks on YouTube. I have recommended this to a colleague already this morning. These don't have the physical health benefits of going for a walk in nature, but they do have reasonably good mental health benefits. So it can be good if you can't go out for whatever reason. I would particularly recommend the 4K relaxation channel on YouTube, which has got basically an infinite variety of walks as far as I can tell. Okay, secondly, you might want to work on identifying and accepting your emotions. So mindful practices like meditation are helpful in stressful waits, but because mindfulness draws attention to your current state, sometimes it can backfire. So if you're feeling lonely, for example, it can actually make you feel more lonely because now you're more aware of it. In a stressful wait, distracting yourself effectively can actually be more helpful than mindfulness. I will talk about that next. And in the meantime, go into this in a little more detail. So I've just casually put that up there. Work on identifying and accepting your emotions, which is incredibly difficult for, I think, a lot of people, maybe all of us. I personally often have no idea what specific emotion I'm feeling, and in some cases I find them really overwhelming and even scary. So here I'm just going to step away from the science for a moment and tell you a couple of things that have worked for me. I don't know whether they will work for others, but they are harmless things to try that are worth a go. So first of all, the classic breathing GIF. So you would find this just by Googling breathing GIF. And this is generally my first step if I'm feeling overwhelmed, is to try and slow my breathing down, because I can end up going quite a lot, which is, I mean, it's not a deep breath because I'm breathing into sort of the top of my chest rather than using my diaphragm effectively to pull down the air as much as possible. Breathing in this rhythm kind of helps you to breathe in more deeply, as far as I can tell. It's quite difficult to not fill your lungs entirely if you're breathing this slowly. If you don't like a breathing GIF, then just slowly tapping your fingers 1, 2, 3, 4, 5 can help with establishing a similar rhythm. Okay. The other thing that can help me with overwhelming emotions is a sensory grounding exercise. So thinking about what I can see, hear, touch, smell, and taste. And that helps me be aware of the here and now in terms of what I'm sensing, and it takes my attention away from my emotions. That often for me means that they don't feel quite so overwhelming. So to do this, you would simply name 5 things you can see, 4 things you can hear, 3 things you can touch, 2 things you can smell, and 1 thing you can taste. So sometimes the answer is going to be nothing. That's okay. The important thing is you're bringing your attention to your current experience. You might also like to rearrange the order of the senses so it works better for you. So together, slow and deep breathing and concentrating on what I'm experiencing usually get me out of intense or confusing emotion and help me decide what I want to do with my emotions. This kind of thing has taken me years to learn, and I'm still not very good at it. So if it's the kind of thing you try and go, what, that didn't help at all, A, please don't be discouraged. Give it a few tries before you decide it's not the right thing for you. And B, remember that there are other techniques out there that you can use, like body scanning, for example, which is just sort of mentally running through the different parts of your body and allowing yourself to sense what's going on in them. Yeah, there are lots of techniques out there. This is a very quick run through of this. So, mindfulness. I talked about distracting yourself efficiently. So the best way to do that appears to be finding something that will put you in a state of flow. And I'm going to resume the research-based information here, having stepped away from it. So flow is being intensely and enjoyably absorbed in a task to the extent that you don't notice the passage of time. So just in case you are in a lockdown or go back into a lockdown or a quarantine, in the particular case of being in a COVID lockdown, the evidence suggests that it's better to prioritize flow over mindfulness to help you with stress. The thing is, what causes flow seems to be entirely up to the individual. So I experienced it while I was writing this talk, but I've also experienced it playing video games, hill walking, and cooking, which surprised me. Also once when many times when I was working as a bartender. So it's really a huge variety of things that can cause flow. Basically, the necessary components you're going to need are finding an enjoyable task. So not a task that you are looking forward to having done, but a task that you are enjoying in and of itself. You need to be actively doing something, and that could be physically or mentally or both. You've got a goal that's challenging but achievable, and you can tell how your progress is going. So here I've just got like a random list of things that might trigger flow for different people in case you need some inspiration. Designing an experiment, putting on makeup, playing football, doing the gardening, singing, meditation, writing a research paper, training a dog, and lifting weights. So hopefully one of those will have made you think, oh yeah, I like that. There are some things you can do work on that list of examples. One of the nice things about academic work is it does tend to offer quite a lot of opportunities for intensely mentally engaging work. But it can be also helpful to have non-work activities that you know will trigger flow, especially if work is currently causing you stress. Okay. The final thing I want to talk about in terms of tips for coping is meeting your fundamental psychological needs. So I mentioned these can tend to drop when you're stressed earlier. When that happens, when we do manage to meet them, we release less of the cortisol, stress hormone. We have fewer physical symptoms of nervousness as well, and we just sort of generally feel better. So you might want to think about ways in which you can increase these. So connectedness, you're going to want to find some kind of life goal or skill that's related to your community, whoever that is for you. Competence, a life goal or skill that you can track your progress and getting better at. And autonomy would be a life goal or a skill that's related to your personal values. Again, academia tends to offer quite a lot of opportunities to do these things, but you again might want to look to your personal life to find these things, because they can work can get you down, so you need to have things that are not work to help you. All right, since I've just been talking about connectedness and community, supporting others is sometimes really tricky. So this is a lot of information on one slide. I'll come back to it when I'm doing questions, if that will work, or you can ask me for a copy of this slide or the summary that's on the summary sheet if you want. So the intention of most of the actions on both sides of this list is to be helpful, but they might be interpreted as helpful or unhelpful depending on the person and the situation. So a really good example of this is reminding someone of their past success appears both underneath positive reframing, which is likely to be helpful, and unwanted positivity. So when you are giving support to other people, when you want to receive support yourself, it's useful to think for yourself, what do I need? Who might be able to offer that to me? And if it's a person that you are offering support to, you might want to ask them which of these things would be helpful for you. Like, do you want distracting? Do you want practical help? Do you want me to just pretend the whole thing isn't happening so that we don't have to think about it? Okay, last couple of things. Some resources, because that's always good practice when you're talking about mental health. So 3 apps that I like are Calm, Headspace, and Youper. So Calm and Headspace are pretty similar in that they offer mainly meditation, audio or video. Youper also offers that, and mood tracking and mental health support. So it can be helpful if you have a pre-existing mental health problem and you're trying to deal with a stressful weight. And finally, a bit of support just in case you need someone to talk to. Hopefully my talk has not made you go, oh God, I can't cope anymore. But just in case that's happened, Samaritans, Shout, and Mind are all able to offer help with these kinds of things. And the very last thing I want to say is this talk was originally a much longer interactive talk that I offer as part of my freelance work as a science communicator. And I go into a lot more depth and cover a lot more coping techniques. So if you would be interested in a longer session, or you know someone else who would, you can have a look on my website, thatthinkingfeeling.co.uk, under workshops for more information about what those workshops are about. And you can also find the resources for this talk here in case there's anything you missed. That's it.
Thank you. Thank you, Claire. That's brilliant. I'm sure people will benefit from that, particularly at the moment. I know that there's a lot of grant rounds of just your own at the Alzheimer's Society as well. Has that just closed, I think?
No, it's
still
still
open?
open till the, oh God, I've
September.
completely blanked on the date. I feel like it's the 16th of September, but it's that week. I don't think it's that day.
So anybody who's stressing about funding right now, of course, you can go onto the Alzheimer's Society website and have a look there, where there are funding opportunities across the board available at the moment, from PhDs through to fellowships and grant rounds as well.
Yes.
The NIHR calls, I think, are there as well. So hopefully that's not stressing anybody out too much. But I think the strategies you've suggested there are particularly useful, Claire. We also have a number of blogs and articles and podcasts on this same thing as well on our website. So do have a look at dementiaresearcher.nihr.ac.uk. We did have a question before from David Ross, and I apologise, David, for missing this before. I think he asked his question related to how we can implement earlier and better diagnostic systems when we're hampered by slow GP recognition and referral. I think I'm going to have a go at this one first of all. I think one of the big challenges that we still face, and this isn't really particularly related to today's presenters, but of course is encouraging people to seek early help from a GP. Now, I know GPs are so variable in how they'll respond when you do go to see them. The usual practice will be to investigate and to check that there isn't something else going on first, and then refer on to a memory clinic as well. But it's really important that people go to their GPs in the first place. And I think and if you're not getting the response you want, is to continue to push, particularly if you're under 60. I think you probably find it harder to encourage your primary care professional that you really have something going on. So I think is to be persistent and keep going and getting that help. And have a look on the Alzheimer's Society website as well, where there are a whole bunch of other resources to help understand the earliest symptoms and what they can be, because they are so different for each individual, and of course different parts of the country and different parts of the world treat this very differently. Hopefully some of the early diagnostic tools that Coco presented on earlier, and other podcasts and webinars which I mentioned before, looking at gait analysis and blood tests and other types of cognition tests will come in and will make that earlier diagnosis helpful or more possible. I hope that helped answer your question, David. He mentions he's got a book as well, but I won't go into that. Okay, Vito asked for the YouTube link. I've shared that below, but if you go to YouTube and search Dementia Researcher, you'll find us there as well. David also says that from his own experiences, that stress is a major factor, of course, in dementia, especially in chronic long-term stress over decades that has resulted in FTD for his wife and sister. I'm sorry to hear that, David. But you're absolutely right. Anxiety, loneliness, mental health are all identified in the Lancet's report as potential risk factors for Alzheimer's disease, as well as diet and alcohol intake and smoking and pollution and good quality air and exercise, and all these things are mentioned as risk factors. And I think we might come on to some of those as we move through some of our forefront talks. If you have a question for Claire, please post it now. And while I'm just waiting to see if we have a question for Claire, I was going to ask you, so would you say, obviously you've left academia, which isn't necessarily the best advert for
No, it was right for me. But basically, I think if someone had told me this kind of feeling is normal while I was still in academia, I would probably have sought help a lot earlier. So that's a large part of what I'm trying to do now, is to encourage people to seek that help if they need it, and to know coping mechanisms for themselves, because I don't want people to go through the burnout that happened to me.
No, of course. And you're a great advertisement for demonstrating how people can still use what they learned in academia remain in the field, stay associated with the science or the particular disease or area of discovery that they're passionate about, but in a different way.
Yeah,
Um,
definitely.
and in that vein, of course, I think there are positions advertised at the Alzheimer's Society at the moment in your PR teams and communications teams and
Yes,
outreach.
I'm not sure if the advert is still open, but if you want to be my manager while my actual manager's on maternity leave, then we are looking for someone now, or we have been recently. So.
Okay, well, it doesn't look like we're going to get any questions for you, I'm afraid. But don't worry, I really enjoyed your talk, and I'm sure others will
Thank you.
too, as they've hopefully watching this back on YouTube as well.
Yeah.
So thank you very much. Coco had to jump off, I'm afraid,
She
so we
did,
haven't got
yeah.
her back with us. But we're really grateful to both you, Claire and Coco, for joining us today, and Alzheimer's Society for working with us on these particular podcasts. Not podcasts. I do too many podcasts on this webinar. We have profiles on both of our speakers from today on our website. So if you, which includes details of their Twitter accounts. So if you'd like to find out more about them and their work and their Twitter, and Claire's written a couple of blogs for us too, you can find those on our website at dementiaresearcher.nihr.ac.uk. And I'm sure Claire's work will also be on Alzheimer's Society's website as well, which is alzheimers.org.uk. Our next webinar comes on the 24th of September, where we're going to be hearing speakers from University of Stirling and Bangor University as well, on redesigning care homes to improve well-being of people living with dementia. And our career session on that day will be about parenthood and academia, something that I think might have been useful in July as school holidays start. But schools will be back by then, so hopefully that'll help you get through the half term. Then in October, we have Soyeon Hong, who's going to be talking about from the UK Dementia Research Institute at UCL, and Dr. Julia Ravi from Alzheimer's Society, who are going to be talking about microglia and synapses loss in neurological disease, and using in the careers talk, we'll be using social media for science communications. Then in November, we have Dr. Gemma Lace from the University of Salford, and Cherie Nyota, and Anna Smith from Alzheimer's Society, who are going to be talking about brain waste disposal mechanisms in dementia and involving people living with dementia in research. So some good patient and public involvement. And then our last talk in this series will come on the 17th of December with Dr. Melanie Handley from University of Hertfordshire, and Claire will be back with us again. And we're going to be talking about person-centered care in hospitals and how to write for different audiences, which I think is particularly helpful for academics who might be writing for a public audience or an academic one. So thank you very much, everybody who's found time to join us today, and everybody who is watching this back on YouTube. Thank you very much for all your lovely comments in the comments box as well. Gary Hodge there has said he really enjoyed the talks. Lots to think about. So we hope this has inspired you in your own work and has been helpful in your career. And we'll look forward to seeing you all again at the end of September, on the 24th of September, for our next Alzheimer's Society Forefront webinar. Thank you very much.